Our world is a symphony of stimuli, both the external five senses, light, sound, taste, touch, and smell, as well as all the internal stimuli that are essential in the maintenance of homeostasis, a constant optimum internal environment. But how do we perceive these stimuli, or changes in specific conditions? It all begins with specialized cells called receptor cells, the sentinels of our sensory systems. These remarkable cells possess unique sodium channels that act as gateways, controlling the flow of sodium ions. Imagine these channels as tiny, highly selective doors, poised to swing open in response to specific stimuli. Some are opened by application of pressure, such as the Pacinian corpuscles, present in the skin dermal layer, while thermoreceptors have sodium channels opened by change in temperature. Photoreceptors' sodium channels are opened by alterations of light intensity. The list is long, and we humans do not have the full range of receptors present in other animals. Those are covered in a different video. This specificity of what it takes to open sodium channels of receptor cells is key to preventing the overload of information by uncontrolled bombardment of stimuli. This could cause the gridlock of our relay neurons in the brain and spinal cord, preventing a clear awareness of the external world around us and the internal environment of our bodies. An awareness that probably represents the single most important tool in the survival kit of animals. So, different receptor cells have sodium channels opened by different stimuli. But how does this result in us becoming aware of sounds, tastes, smells, and so on? Well, the difference in the number of sodium ions in O&D outside of the receptor cells produces a potential difference, or voltage. Therefore, once the sodium channel gates are opened by the stimulus, the sodium ions will flow along the voltage, producing a current. This is the language of the nervous system, the means by which information travels throughout our bodies. Once a receptor cell has transformed a stimulus into an electrical signal, this signal must be transmitted to the brain for processing. This is where neurons, the fundamental units of the nervous system, come into play. Neurons are like the wires of an intricate circuit, carrying messages throughout the body. Unlike receptor cells, which rely on a variety of stimuli to open their sodium channels, neurons utilize voltage-gated channels. These channels are highly sensitive to changes in electrical potential across the neuron's membrane. When the voltage reaches a certain threshold, all the sodium channels swing open by a process of positive feedback, allowing a full influx of sodium ions. Let's take a more detailed look at this stage. The all-or-nothing law is an essential control mechanism that prevents the central nervous system, made up of the brain and the spinal cord, being overwhelmed by an excess number of action potentials. A good analogy of this principle is a light switch. It can be either on or off, but nothing in between. Similarly, a neuron, sensory, relay, or motor either fires an action potential, a brief electrical impulse, or it doesn't. There's no partial firing. This principle is governed by the neuron's critical threshold. If the stimulus is strong enough to depolarize the neuron to this threshold, all the voltage-gated sodium channels in the neuron are opened by positive feedback, and a full action potential is generated racing down the neuron's axon. If the stimulus falls short, no voltage-gated channel is opened. Nothing happens. The neuron remains at rest, its electrical potential carefully maintained. Depolarization of the axon due to inflow of sodium ions is followed by repolarization caused by the outflow of potassium ions out of the axon. Repolarization will then cause depolarization in the next section of the axon. This process continues until the action potential reaches the end of the axon. What happens then? Bridging the gap signals across synapses. Neurons, though intricately connected, do not physically touch each other. Instead, they communicate across tiny gaps called synapses. When an action potential reaches the end of a neuron's axon, it triggers the release of chemical messengers called neurotransmitters. These neurotransmitters diffuse across the synapse and bind to ligand-gated receptors on the postsynaptic neuron, each receptor being specific to a neurotransmitter, much like keys fitting into locks. Some neurotransmitters are excitatory, increasing the likelihood of the generation of an action potential, such as noradrenaline, serotonin, or dopamine. while others are inhibitory, such as gamma-aminobutyric acid, or GABA for short, which act to inhibit or suppress production of an action potential in the receiving neuron. The combination of these different types of neurotransmitters fine-tune the likelihood of generating an action potential, the postsynaptic neuron. We will look at neurotransmitters and effectors in greater detail in future videos. Meanwhile, if you like this video, don't forget to hit the like button and subscribe to this channel and press the notifications bell. And of course, most importantly, don't forget to stay curious.